The French army was winning. King Charles VIII had marched 20,000 soldiers over the Alps, through the Italian city-states, and straight into Naples without much resistance. The Pope had fled. The local nobility was cooperating. By every military metric, this was going extremely well. And then his soldiers started falling apart. Not from battle wounds, not from starvation, from something nobody had a word for: lesions, spreading sores, bone pain so severe that men who had survived actual warfare were begging for death. Contemporary accounts describe the skin peeling, the smell, the speed of it. Within months, Charles VIII had disbanded his army, not for political reasons. His army had physically ceased to function. The disease that destroyed it would eventually be called syphilis. And for the next 5 centuries, historians and doctors and scientists would argue about where it came from. The standard answer, the one you probably learned, is Columbus. 1492, he lands in the Caribbean. 1493, his sailors return to Spain. 1495, the disease explodes in Naples. Neat timeline, clear causation. Except, in early 2024, a team of geneticists published a paper that made that timeline significantly more complicated. And in January 2026, Science published a commentary that flipped the entire narrative on its head. And what they found was, depending on your perspective, either reassuring or deeply unsettling. First, let me explain why the Columbus theory was always shakier than it sounds. The theory is called the Columbian hypothesis, and it has a lot going for it. The timing is genuinely striking. The skeletal evidence from pre-1493 European burials is mostly clean, very few signs of treponemal infection, which is the bacterial family that includes syphilis. Post-1493 skeletons tell a different story, and pre-Columbian skeletons from the Americas do show the telltale bone lesions. So the bacteria was clearly present there before Columbus arrived. But here is the complication that the hypothesis has always struggled with. Treponema pallidum, the bacterium that causes syphilis, doesn't work alone. It's one member of a family. That family includes the bacteria behind yaws, a tropical skin disease spread through casual contact, Begel, a bone disease common in arid regions, and Pinta, a skin condition found in Central America. These 4 diseases, syphilis, yaws, Begel, Pinta, are so genetically similar that for decades, paleopathologists couldn't distinguish them reliably from bone lesions alone, which meant every time someone found pre-1493 European skeletal evidence that looked treponemal, the debate exploded. Is this syphilis? Or is it yaws that arrived through African trade routes in the 12th century? Or is it Begel? Nobody could prove anything, because nobody had ancient DNA. Until 2024, and until January 2026, when science confirmed the answer had been found. A paper published in Nature in early 2024 did something that wasn't possible 10 years ago. The researchers took skeletal remains from multiple archaeological sites across the Americas, some dating back over 5,500 years, and successfully extracted and sequenced ancient treponemal DNA. This is genuinely hard science. Ancient DNA degrades. Contamination is a constant risk. The fact that this worked at all is a technical achievement. What they found was a phylogenetic tree, an evolutionary family tree for the entire Treponema family. And the picture it showed was not what anyone expected. The most ancient strains they found were not in the Caribbean. They were in the interior of South America, Colombia, the Amazon basin. Sites with no documented European contact. And these ancient strains were the direct ancestors, the evolutionary predecessors of the strain that eventually became syphilis. But here's the thing. In January 2026, the journal Science published a perspective commentary on the discovery. and it contained something that most news headlines missed. The genome they found wasn't just ancient syphilis. It was a previously undescribed subspecies of Treponema pallidum that had been circulating in South America at least 3,000 years earlier than any known ancient treponemal genomes. It was closer to the root of the evolutionary tree than anything we'd ever found before. The Treponema family was not new. It had been in the Americas for thousands of years before Columbus. The strain that became sexually transmitted syphilis evolved gradually from strains that were skin contact diseases passed between people casually through touch, not sex. So here's what the evidence now suggests happened. Columbus's sailors didn't import a disease that already existed and was thriving in a Caribbean population. They encountered a bacterium that had been quietly evolving for millennia, made contact with a strain their immune systems had never seen, brought it home, and then that strain, which may have been more virulent due to the new host population, encountered European bodies with zero immunity. Not an arrival, an introduction between two populations that had been evolving separately for thousands of years. The disease didn't come from nowhere. It came from a very long somewhere. Here is where it gets properly strange. When you build that phylogenetic tree and keep going backwards past the 5,500-year-old Columbian skeletons, past any human civilization, you find something unexpected. Closely related treponemal bacteria infect non-human primates, gorillas, baboons. Some of the same species that ancestral humans were sharing habitat with hundreds of thousands of years ago. The current scientific consensus is that Treponema crossed from primates into early humans, probably multiple times, probably through close contact in shared environments, long before anything we'd recognize as civilization, long before farming, long before pottery. What eventually became syphilis started as a skin infection in a primate. Then it crossed to early humans. Then it evolved alongside human populations for tens of thousands of years. The sexually transmitted form appears to be a relatively recent evolutionary adaptation, recent meaning, in geological terms, sometime in the last several thousand years. So 1495 in Naples wasn't the beginning of the story. It wasn't even close to the beginning. It was one specific chapter in a story that starts with a primate in a forest, long before the first human being had any concept of what a disease was. That reframe matters, because the question isn't who brought this disease to whom. The question is, what conditions caused a bacterium that had been quietly circulating for millennia to suddenly become catastrophic? And the answer to that question is 20,000 malnourished, immunocompromised, overcrowded soldiers in a city that was not prepared for them. The bacteria didn't change in 1495. The conditions did. Now I want to zoom out, because syphilis gets all the drama and the other STIs have equally strange origin stories. Gonorrhea. Gonorrhea is genuinely ancient in a way that syphilis might not be. The Egyptian Ebers papyrus, written around 1550 BCE, but probably compiled from much older texts, describes urethral discharge in language that medical historians are fairly confident is gonorrhea. Galen named it in the 2nd century CE, from the Greek for flow of seed, because he thought he knew what it was, and he was completely wrong. The book of Leviticus contains what reads, to a modern epidemiologist, like a public health protocol for managing exactly this kind of infection. Gonorrhea has been with us for at least 3,500 years of documented history. Genetic analysis pushes the actual bacterial lineage back much further. Neisseria gonorrheae. The bacterium appears to have been infecting humans before agriculture, before cities, before most of what we think of as civilization. It co-evolved with us, gradually becoming more adapted to human transmission, getting better over millennia. at doing exactly what it does. And despite being one of the most common bacterial infections in human history, nobody correctly identified what it was until 1879. That's when Albert Neisser, the bacterium is named after him, isolated it under a microscope and proved it was its own distinct pathogen. Before that, for most of recorded medical history, gonorrhea and syphilis were believed to be the same disease, different stages of the same infection. In 1767, a surgeon named John Hunter decided to test this by inoculating himself with what he believed was gonorrheal discharge. He developed syphilis, concluded the diseases were identical, influenced medical thinking for decades. The sample had contained both. Hunter was brilliant, rigorous, completely wrong, and set back the field by at least a generation. His portrait hangs in medical schools across the world. This is the part of the video where I'm supposed to sell you something, and I'm going to, but I want to make the case for why it's actually relevant here, rather than just pretending it fits. The entire reason we know any of this, the 5,500-year-old DNA, the phylogenetic trees, the primate connection, and now the fact that we can distinguish between subspecies with incredible precision, is because of advances in genetic testing that didn't exist 15 years ago. In April 2026, a study came out that developed a new typing scheme for Treponema pallidum based on 7 genes. They tested it on 542 samples from 31 countries and got 82 different genotypes. And guess what they found? The level of genetic diversity was equally high across all subspecies: syphilis, yaws, Bagel. The bacterium wasn't simple. It was complex and diverse this entire time. The same technology, DNA sequencing, searching for specific genetic markers, is the science behind modern STI testing. And here's the thing. The single biggest factor in STI transmission across all of human history, from the Egyptian papyrus to Naples in 1495 to today, is not immorality, not promiscuity, not any of the things these diseases get associated with. It's not knowing you're infected. Gonorrhea, in particular, is asymptomatic in a significant percentage of cases. You can carry it, transmit it, for months without any indication something is happening. Same with chlamydia, which I'll get to in a second, so the product I'm linking in the description is an at-home STI test kit, specifically MyLab box, which tests for multiple infections. You do it at home, send it to a lab, get results in a few days. No awkward appointment, no clinic waiting room. I think the reason to mention it in a video literally about the 5,000-year history of these diseases is that the most significant difference between 1495 and now isn't the medicine. It's that now you can actually know. Link in the description. Onward. Right, chlamydia. Chlamydia trachomatis, the sexually transmitted bacterium, is part of a larger family called Chlamydiaceae, and the evolutionary history of this family is almost absurdly old. Non-sexually transmitted chlamydia species infect fish, birds, reptiles, amphibians. The bacterial lineage that eventually produced Chlamydia trachomatis has been infecting vertebrates for hundreds of millions of years. Chlamydia trachomatis itself, the specific strain that infects humans, appears to have diverged from related strains somewhere between 50,000 and 700,000 years ago. The estimates vary depending on the methodology. But the conservative end of that range is still before Homo sapiens as a species fully existed. This is not a disease that humans caught from somewhere. This is a disease that evolved with hominids. It adapted to us, or something very close to us, before we were fully us. The ancient Egyptians had a word for trachoma, a related chlamydia infection affecting the eyes, in texts dating back to 1500 BCE. but the sexually transmitted strain is almost certainly older than any written language. These bacteria are not passengers in human history. They are, in a very uncomfortable sense, participants in human evolution. They shaped our immune systems. Our immune systems shaped them. This has been going on for longer than modern humans have existed. So, back to Naples. 1495. The French called the disease Italian. The Italians called it French. The Poles called it German. The Japanese called it Chinese. Everyone found a foreigner to assign it to, and the assignment always told you more about existing prejudices than about actual microbiology. This pattern has never stopped. The 1918 flu was called Spanish because Spain was the only country not at war, and therefore the only country with a free press that could report on an epidemic. It probably didn't start in Spain. The Spanish just weren't censoring their newspapers. The AIDS epidemic in the 1980s followed the same arc. Blame the gay community. Blame Haitians. Blame Africa. The moral categorization of victims delayed funding, delayed research, delayed acknowledgement that this was a public health emergency affecting everyone. The instinct to locate disease in the other, in the stranger, in the morally suspect, is itself one of the most consistent features of recorded human history, and it has caused, across centuries, at minimum, as much suffering as the diseases themselves. What the ancient DNA science shows us is that these diseases don't have a moral origin. They have an evolutionary one. They predate morality. They predate civilization. In some cases, they predate the human species. They are not punishments. They're extremely old tenants. So where did STIs come from? Chlamydia, older than Homo sapiens, evolved with hominids, has been infecting things with backbone since before the dinosaurs went extinct in its more distant forms. Gonorrhea, older than farming, documented in Egyptian medicine 3,500 years ago, co-evolved with humans for long enough that we may never know the actual start date. Syphilis, the youngest of the three, and the most complicated, almost certainly connected to the Americas and to the Columbian Exchange. But the 2024 evidence confirmed by the commentary in Science in January 2026 shows that the story is less Columbus imported a disease and more two populations that had evolved separately for millennia finally met, and one of them had an ancient bacterial companion the other had never encountered. None of them came from nowhere. All of them came from deep evolutionary time, and all of them in every era prompted humans to ask the wrong question first. Not, what is this? And how does it spread? But whose fault is this? The bacteria, for what it's worth, did not care. It just kept evolving, and today, here's where it gets properly interesting. In May 2026, scientists published the first ever study where artificial intelligence was used to model the structure of 99% of all proteins in Treponema pallidum. They identified 63 proteins potentially linked to pathogenicity and found new targets for a vaccine. The bacterium that has been with us for thousands of years is finally starting to reveal its deepest secrets. We've been asking, where did it come from for 5 centuries? And now, thanks to ancient DNA and artificial intelligence, we don't just know where it came from, we're starting to understand how to stop it. But that's a story for another time.